So I think we ended talking about translation, transcription. Now, for the exam, questions will be on this. This is the kind of stuff that, you know, could be even like a written question as we kind of discuss and see if you kind of understand it. If you understood what we talked about previously and can go over the notes, and you'll see that I will post some videos online as well about it so you can review. And I'm gonna have some more Learn Smarts to do in chapter 3 and 4 for you guys to do as an assignment, right? They'll probably be due that day we are going to have our exam. So I want those assignments kind of done before the exam date, so then you're not done with the exam and then have to do assignments. Kind of defeats the purpose, right? We want to be able to do the assignments to prepare for the exam. So I will release those, and you will have until the, I believe, the 24th to get those done. So let's take a look at what we mean by regulation of gene expressions right here. And with the regulation of gene expression, we kind of mentioned that just as important than how to make it is when to make it. So when we discuss all of these kind of highlighted material right here, I would want you guys to focus in on those material. So everything but red blood cells, everything but your egg and your sperm cells are going to have the same complement of DNA. So the DNA that we find in our cheek cell, that's readily accessible. The DNA that we find in your stratified squamous cells of your epidermis, all of those cells have this exact same DNA as any deeper located cell, like your white blood cell, like your stem cells that are very important, right? So just keep that in mind. Same DNA. So we can basically ensure that if I check one cell, it doesn't matter where that cell's found. As long as they have the full allotment, it should be identical to every other cell that has that same allotment. And I think we did mention this about getting that certain genes are turned on in some cells, while in other cells they are turned off. So then you can have neighboring cells that produces hormones like insulin, and then neighboring cells next to them will produce a hormone like glucagon that does the exact opposite. Now, let's talk about the cell's life cycle. What happens to the cell, and when does it undergo certain processes? Keep in mind that unlike when we mentioned protein synthesis, that's going to be done continuously throughout the lifespan of a cell. I wish that they would make this so we can have the whiteboard a little bit more accessible, but what I mean is, unlike protein synthesis, the next kind of situation we're going to discuss is called DNA replication. DNA replication only occurs at set times, and that's it. Protein synthesis is going to occur just about all the time. You're going to need more proteins, you're going to need more receptors, you're going to need more just about everything. So when you take a look at this cell life cycle, you'll notice that it's not quite split into 4 parts. Not equally. This middle part right here, S phase, is much longer than the other parts that will kind of entail the lifespan of a normal cell. So what do we see? We've just produced that cell. As we just produced that cell, a brand new cell. That brand new cell will then undergo this G phase, G1 phase. That's when we start to reproduce more and more nucleic acids, more enzymes, more energy. Because remember, in order for a body to grow, in order to have growth and repair in the body, we need to undergo cell division. So then one cell splits into 2. But the cell division part is just a very small part of the normal life cycle of a cell. So the rest of the parts lead us to the cell division phase, or mitosis. So what we see is this: the G1 phase, routine metabolism, production of the nucleotides that will be required for once we have DNA replication. You're going to need more energy, more ATPs. So you need to double a lot at this point. And the cell's going to grow. The cell's going to then get ready for the next phase. The next phase in that cell's life cycle is the S phase, or synthesis phase. That synthesis phase is where we will see another cellular process called DNA replication. Now, DNA replication is actually slightly easier in some ways than protein synthesis. It's just one step in terms of we duplicate everything. Remember, the whole concept is this. You want to double everything. If you think about it, you're going to have one cell become two. So everything needs to be doubled. So then when we split in half, that original cell looks like the two cells you reproduce from it. Perfect. So in mitosis, we have this, we have this DNA replication phase occurring in the S phase, and then we are going to go to the next gap phase, or G2 phase. This G2 phase then gets us ready for mitosis. Right? So mitosis is the actual process where we've already duplicated the DNA. Now we have twice as many chromosomes, and at this point, we've doubled just about everything else. The organelles are doubled, and what we're going to do at this point is now start splitting that cell in half. Think of this cell as being just larger, and now we're going to split it right in half. The cell division phase starts with prophase, metaphase, anaphase, then telophase. That last phase where you completely separate the two cells is called cytokinesis. So once we undergo cytokinesis, we have two cells that we made from one. Those two cells are identical to each other in every way. If they're not identical, we have a real issue. Now, those two cells that we produced was also identical to the original strand or original cell that started the process. Again, cytokinesis is when we literally tear the cells apart and form two cells. Now, this is replication of DNA. DNA replication, we're going to double the DNA. It happens to all 46 chromosomes. And with all 46 chromosomes, we're going to add a second arm to them. So we're going to start off with two parent strands. And out of those two parent strands, right, so two parent strands for one chromosome. Out of those two parent strands, we are going to produce 4. We're going to double everything. But the weird thing is this. Because of how we count the chromosomes, they don't count as doubling. In other words, right, we're going to start off with 2n number of chromosomes, with an n being 23 chromosomes. So when we started, we have 46 chromosomes. 2n, 2 times 23, right? 46 chromosomes. But when we are done with DNA replication, we still have 2n chromosomes. And it can get confusing, but remember when I said when you have two chromosomes that are attached to each other, they count as one. As soon as they detach, they count as separate chromosomes. So whenever you see a chromosome with 2 arms, like this. So here's a chromosome with 2 arms right here. When you see those chromosomes with 2 arms, what happens? Well, they count as 1 chromosome, even though you've undergone DNA replication and you should have doubled it. But since they're attached, they only count as 1. As soon as we split it apart, what happens? They count as separate chromosomes. So it's kind of weird. It's just the way that they kind of write it that makes it confusing. So let's talk about DNA replication first and what it means for the semi-conservative replication. Now, when you're doing more molecular biology, I'm pretty sure you guys will have a lot more detail about this. Sometimes, especially when we start discussing as well, if you're in molecular biology, you'll also get through this as well in a lot more detail. So keep that in mind that we are not going to the same detail as you might